Tools is that it enables what I call multi-use metabolic databases. So once we've created a metabolic database like HumanPsych, the different modules of Pathway Tools let you use HumanPsych for many different purposes. It's an online encyclopedia that you can go to and look up information about human pathways and reactions and metabolites. It also has a zoomable metabolic map diagram that looks a lot like Google Maps, and that diagram can be used for omics data analysis, and there are several other omics data analysis tools available. It's also a queryable database that you can ask queries to, like find me all the enzymes that are regulated by ATP and have a given molecular weight range or PI range. You can issue structured queries to the database, and you can develop metabolic flux models from the databases. But let me now focus in on SmartTables, which is a relatively new component of Pathway Tools. We used to call SmartTables web groups, but people thought it was some kind of social networking site or something, so we've just changed the name in the new version that we've released. And although I'll focus today on the metabolite-related aspects of SmartTables, SmartTables actually can manage many different types of objects. They can manage sets of genes, of pathways, of sequence regions, and for each type of object, there are different types of manipulations provided in SmartTables. Also, SmartTables can be applied to any database that you have a Pathway Tools database for. And I'll illustrate it with the human database, but again, you could use it for E. coli or yeast or whatever organism you care to use it for. What SmartTables let you do is collect and store within your web account lists of database objects and then perform many manipulations. You can transform those lists of objects into related objects, like transforming a gene list into the list of pathways that those genes are involved in. You can filter and combine SmartTables, like by taking the union or intersection or set difference of two or more tables. You can share them. You can make them, initially they're private, you can make them public, or you can share them with specific collaborators. You can export them to a spreadsheet file, and you can perform omics data analyses with them. So let's get into these in more detail. You access SmartTables through this main menu at the top of the website. Again, it's available at all our different websites. And be aware that the best way to create and save a SmartTable is to create an account on BioPsych or to set up an account on your own locally running version of Pathway Tools and then save SmartTables within your account. How do you create a SmartTable initially? There are several different ways. You can simply type in the names of metabolites or genes or pathways that you want to have in the SmartTable that you're creating, or you can create them from search results. You'll see in the search result pages, you'll see create a SmartTable from these results in various web pages. And you can upload a tab-delimited text file of metabolites or genes to create a SmartTable that way. People always ask, well, what exactly is in that tab-delimited file? And the answer is it's a list of metabolite names or identifiers using our internal identifiers. In the future, we're going to create a metabolite mapping service that will help people transform lists of metabolites with various different identifiers like KEG identifiers and KEBI identifiers into our metabolite identifiers, but right now the importer doesn't recognize other identifiers. So here's what a newly created SmartTable looks like. Is it's simply a table of one column where the first column is the database objects from a specific pathway genome database that are defined in your table. So one thing to keep in mind is that every SmartTable is associated with one specific database, be it E. coli or human or whatever. Okay, so once we've created a SmartTable, a number of different menus appear above the SmartTable on the web page, and one simple thing you can do is add additional columns to the SmartTable. Now you can actually, when you load in a data file, the data file can contain arbitrary additional columns of data. So that's one way to define additional columns in a SmartTable. But another way to define additional columns is by showing attributes that are already defined in the database for each of these objects. So for example, I can ask it to show the IDs of these compounds that we have stored in our database. We have Kebi IDs and KEG IDs for many compounds. We don't have that many HMDB links so far, but we're working on that. So this is one simple type of property you can add. You can also add molecular weight and monoisotopic molecular weight as additional properties. You can also add SMILE strings or INCHI strings are also present in our databases, and the chemical structure is present in our databases. So those are additional columns that you can add, additional properties. And we have information on regulation of metabolism by these metabolites. So you can add information about what enzymes a given metabolite regulates and what's the mechanism of regulation. Now there's another type of column that can be added, which is based on what we call transformations. Transformations are new columns that are created by computing from existing columns. So for example, and that transformations come from this other menu, given our menu of our column of metabolites, we can ask it to add new transformation columns by choosing from a menu. And one of the options in the menu is to show all the reactions that that metabolite is involved in. We can also ask it to add a column showing all the metabolic pathways that metabolite is involved in. So here's a simple way to see for each metabolite in your SmartTable, how is it involved in metabolism. We can add in an additional column, which are the genes involved in the pathway. But to do that, we need to select the pathway column as our current column. That'll give us a different set of transformations available, namely transformations on pathways. So here I've selected from a menu of pathway transformations, of which one transformation is show me all the genes involved in that pathway. Now another thing I can do is at any time I can create a new SmartTable from the entire contents of one column. So I've selected this pathways column again, and by clicking on the plus sign I can say create me a new SmartTable consisting of the union without duplicates of all the pathways in that column. So that's shown here. Now we have a new SmartTable consisting of pathways. And again, I can do transformations on that pathway set. I can say show me all the genes in each pathway and show me all the metabolites in that metabolic pathway. So that's a summary of transformations. So there are a number of other operations available on SmartTables. We can take the set union or intersection or difference among SmartTables. We can filter SmartTables by removing or keeping rows based on searches. As I said, we can share SmartTables with specific users by email address or with the public. And we can freeze a SmartTable for publication. We're thinking people may find it useful to publish results as a SmartTable that's publicly available and create an archival record that way. So let's talk about some of the OMICS analysis methods that are available through SmartTables. You're probably familiar with enrichment analysis. It's a statistical method that will find classes of objects that have more or fewer objects present in a data set that than you would expect by chance. So for example, given a list of differentially occurring metabolites from a metabolomics experiment, you can apply enrichment analysis to ask, are there more metabolites in that metabolite set than you'd expect by chance from specific metabolic pathways? And so so in that set of menus that show up above a SmartTable, there's a menu for enrichment analysis, and you can simply select for a SmartTable of metabolites, ask it to perform an enrichment analysis to find the highly enriched pathways. And so I've applied that enrichment analysis to that same metabolite set that we started with, and it creates a new SmartTable whose first column is pathways or pathway classes from our pathway ontology, which I'll explain in a moment. Second column is the p-value from the statistical analysis. And the third column lists the metabolites that are associated in the data set with that pathway, meaning these metabolites are substrates of reactions in the pathway. And so the most highly enriched pathway was NAD de novo biosynthesis, and these 4 metabolites were present in that pathway. It's sorting them by p-value. And you can see several other pathways listed. Now, I mentioned pathway classes. NAD biosynthesis is a class or a category of pathways in our pathway ontology present in our pathway genome database. And so it's showing us that this general category of NAD biosynthesis pathways is also highly enriched for. There are other pathways in that category. So here's another thing that can be done with a SmartTable is there's a command that says paint the SmartTable results onto what we call the cellular overview. The cellular overview is an organism-specific metabolic map diagram that our software automatically generates from the pathway genome database. This is the human metabolic overview, and what you're seeing is individual metabolic pathways. The little dots are metabolites, the lines are reactions. Metabolites are grouped into related classes according to the pathway ontology. So all biosynthetic pathways are on the left. All the amino acid biosynthetic pathways will be grouped into one area. And when you paint a metabolomics data set onto this diagram, there are 2 ways to do it. What we're showing here is a Boolean painting where it's simply coloring in green, and you'll see it better on this version. I don't know if you can actually tell. There's a green one right there. So that was one of the metabolites in our set. It's coloring in green all the metabolites that were in our set in the SmartTable so that you can see the pathway distribution of these metabolites visually to see, do they cluster into specific pathways. And you can zoom in on this diagram further using those zoom controls like Google Maps to see, well, here's a set of related pathways involved in tryptophan degradation and conversion to serotonin, and there were several metabolites from my SmartTable in this super pathway. Now, the second way to paint the quantitative data onto this diagram is, the second way to paint a metabolite set onto the diagram is if there are quantitative expression values or measurement values associated with the metabolites. And those can be inputted, as I said, when you load in the SmartTable from a tab delimited file, you can have additional columns in the file that supply those quantitative data. And they can be really any numbers. What the software does is to map those numbers to a color spectrum and then color the metabolites with colors indicating their expression values. And so they all look kind of similar here, but there are different shades of orange that are being shown here. And when there are multiple columns present in your data file, it will produce an animation. So the live version of this diagram, every few seconds it cycles through a different column, and so the colors change to reflect the expression levels of those metabolites at different time points or for different samples, whatever your columns indicate. And we can also drill down using this same tool to mouse over a metabolite. It will identify what the metabolite is, and you can ask it to graph the metabolite measurements for in this case, there are 4 different time points present for citrate or acetoacetate, and you can show these things as bar graphs or heat maps or XY plots. And here we're zooming in on the diagram a little bit more to show these pathways at a higher level of detail. So you can zoom into your data and drill down on specific values in the data set. One of the things we'll be adding soon is the ability to do these graphs for all metabolites in a pathway. Well, let me acknowledge my co-workers at SRI and various collaborators on these different database projects, and thank the NIH, which funded us, and be happy to take some questions.
Very exciting presentation. Thank you. Well, I have a question in relation of some, things. Basically, correct me if I'm wrong, but you operate within the canonical pathways.
So we have curated in MetaCyc and the other databases, yes, canonically defined pathways from the literature.
Right. What would be your plan to introduce the pathways which are going to be discovered today, tomorrow, etc.?
Really, the plan is to continue on the literature curation.
Right.
So that's you really need for these databases. You want to have an up-to-date, accurate picture of the latest biology. And so for our E. coli database and MetaCyc database, we have curators who are actively scanning the literature for new information and incorporating those pathways. Alternatively, when groups install our software locally, they can update the databases themselves.
Right, and that would be their responsibility. And
yeah,
and all the consequences.
Right.
Okay. Yeah, last question. The last one is, where will you put, in a higher organism as a human sick, where will you put the border between endogenous and exogenous metabolites? Including this gut microbiota things, etc., where it goes?
Yeah, I'll note that a couple years ago there was a biological research project at SRI involving study of nicotine, and so we collaborated with those folks and added a nicotine degradation pathway to human psych. So there's no reason one couldn't include such pathways. Okay. And yes, I think we need an effort to create a gut microbiome database and be able to create joint models of the gut microbiome and the human metabolic pathways. And I'll mention that one of my goals right now is to try to create a center involving the creation of curated human and mouse metabolic databases and gut microbiome databases, as well as metabolic models for those different organisms.
Yeah, thank you.
Fantastic database. Thank you very much.
Thank you.
I have a question on sparse networks or organisms, and I'm thinking in particular of Sean's talk yesterday morning, when you have communities that bring metabolic networks together, and so this very useful tool in filling pathways is very tempting to sort of like create artificial individual organisms that may actually benefit from communities. What are your thoughts on this?
Yeah, in general, a new frontier these days is modeling of microbial communities. And so we'd like to, one of our plans is to expand our software tools to be able to build metabolic models for microbial communities. It's also interesting to think about what kind of data can you gather to constrain those metabolic models. It would be nice, for example, if you could measure the exchange of metabolites between specific organisms in the community. I'm not sure you can actually measure that, but yeah, I think that's a really exciting area.
And I mean, this is very powerful, but also dangerous tool. And I'm thinking yesterday somebody said that we're synthesizing cysteine from serine. Just click on add pathway, there it is. We just can't do that. So what kind of like does the program give us some warnings, limitations?
I'm not sure what you mean by dangerous or warnings or.
I mean, like, it's very easy to create artificial pathways, and it's kind of like exciting to do that. But if it doesn't make sense, it does. Is there an option where it sort of like compares to evolutionary related organisms in order to create warnings?
That's not. I mean, we do build in a lot of checks in the software tools. For example, simply checking for reaction balance is a very straightforward check you want to do. That's one example. We also have checkers for what are called dead-end metabolites, which are metabolites that are only made or only produced by the network, which doesn't make much biological sense most of the time. So yeah, there are many ways that you can check these models for, do they make sense according to some common sense rules of biology? There are probably more types of checks that we could and should add. Be interested in your thoughts on that.
That was just a thought. Thank you very much.
Thank you. Yeah, the software has been applied to draft genomes and to metagenomes. So it will work if you have a genome that consists of contigs, for example. It will handle contigs, and people have used it for metagenome data as well. And there are two ways to do that. You can treat the whole metagenome really as one organism, or if you've binned your metagenome, you can really treat each one as a separate organism. So we should probably move on to the next speaker, kind of over my time here.